All right, yeah, thank you, Anurag, for giving a nice introduction. So what I will do, I will just talk about the current, technology that I'm developing at IIT Ropar. Not developing, in fact, it has been already deployed. And I will discuss how this nanobubble technology is really applicable at commercial scale, how this has a potential, and how this can be a game changer for many of engineering applications. So I'll talk about this technology all throughout my presentation. So first of all, what is called nanobubble? So if you look at carefully the map, there are a different class of bubbles. There are macro bubbles. You might have seen this bubble in your daily life. The bubbles are bigger in size and it rises in the liquid and it bursts on the surface, right? On the other hand, microbubbles are something is milky in nature and it also rises in the liquid, but the size is basically around 1 micron to 2 micron is typically a milky kind of appearance that you'll see. And there are bubbles which cannot be seen by naked eye, and these bubbles are so-called nanobubbles actually, and they are very tiny, very small size. It can't be seen by naked eye, but this can be, you know, you know, this can be seen by a microscope or other instrument, which is a very specialized kind of equipment. By that, we can see this bubble, right? So normally, if I look at the definition of nanobubbles, so nanobubbles are typically, less than 200 nanometer size bubble, which has a very unique property. Uh, you know, so these bubbles are very has a special chemical and physical kind of properties. So we'll discuss these properties in forthcoming slides. So if I talk about the properties of these bubbles, so these bubbles are neutrally buoyant. The buoyancy is not there because the size is much smaller. The size is typically 100 nanometer, and that's why it is neutrally buoyant. It doesn't rise in the liquid. It is a stable kind of bubble. Normally, micro bubble and macro bubble, the bigger bubble, do not has a uh, uh, stable property. I mean, they burst actually, right? On the other hand, if you look at the nanobubbles, they are stable in nature and they last longer in the liquid. They normally last quite longer in the liquid. It has an oxidative property. It can create free radical, and the free radical can be used for degradation of various things, various chemical present in the water. It has also properties called electrochemically active properties. It reduces surface tension, and that's why it also called as a bubble with a cleaning capability. So nanobubble basically reduces the surface tension of nanobubbles, or because of the water, and that basically cleans the surfaces. So it can be also a surface cleaning kind of a stuff, or the normally we use surfactant and chemicals to clean the surfaces. These bubbles can clean without using surfactant. That's the beauty. So without using any chemical, if we are able to clean the surface, that is the one of the biggest property and biggest promising property of the nanobubble. If you look at the chemical engineering applications, it has a high gas transfer efficiency because of smaller size. The surface area becomes very high. Surface to volume ratio becomes very high, and that's why the transfer efficiency is very high. And the transfer rate of oxygen transfer rate becomes pretty good, in the water, and that gives one of the best results, right? It has got, as I said, it's neutrally buoyant. Apart from that, the bubbles are seen to be having a charge, surfaces. They have a charge at the interface, and that also determines the property. If there is charged surface, it can attract the opposite charge particles, and therefore it is really helpful in aggregating the particles or aggregating the contaminants together, right? So these are the typical property of the bubbles, and which leads to a lot of engineering applications, which I will show in my presentation. Um, Looking at the application, if I look at the applications, it has a lot of applications based on the property. So it forms a free radical, and that free radical can be used to degrade micropollutants. Micropollutants are typically chemical or other kind of pollutants. These pollutants can be degraded easily because ROS directly attacks on the molecules, and the molecules can be reduced to a smaller, molecular weight compound. Um, since if you look at the hydroponics in the agriculture, the major problem is basically nutrient transport. So all we do in hydroponics or in other advanced agriculture systems, we supply nutrients via water medium. If you are able to provide better transport, or if you are able to transport the nutrient in a much better way, that can be could be a game changer. That's what we're using here. So if you use nanobubbles, if you're using nanobubbles to transfer nutrient, uh, it gives much better transport of nutrient, and that's why we have seen there is a good transport of nutrient and the better growth of the plant, and also the microorganism like shrimp and fish growing. I mean, if we look at the fish transport or fish growing industries or fisheries, they also require oxygen. Any animals, if you're a waterborne animal, if you are growing any animals in the water, actually, the oxygen demand is there actually, and if the oxygen demand is there, so nanobubble is perhaps the better way of giving oxygen to the bodies, to the water bodies. And if we are able to give the oxygen to the water bodies, we can effectively we can provide oxygen to the animals. So in this way, the fishery, aquaculture, agriculture sector can be catered very well. Similarly, seed germination. If you look at a lot of companies provide seeds actually, and the quality of seed matters actually. So if there is no proper germination of seeds, we will definitely will not see a good rate of production, right? So seed germination is of course the another applications. Environmental application where remediation is there, where wastewater treatment, where we are able to treat the wastewater treatment bodies, water bodies like lake and pond treatment. Particle flotation, as I said, the bubbles are charged in nature, and because of charge, they aggregate the various contaminants like particle present in the water, and they float the aggregation aggregates basically. Water remediation, of course, renewable, as I said, sewage treatment plant, ETPs, of course, in the drinking water sector can be also people are using these days. Anaerobic digestion is one of the application where people are trying to digest, the waste material. If you are using micro aeration, if you are providing nanowables, you can, you can digest the organic matter much faster in presence of, so degradation of organic matter can be enhanced by electron transport. So nanobubble provides a better transport of the electron, and in this way, one can have a better, digestion. Similarly, these days, this algal biomass production is also in a peak. People are finding a new source of food, where algal biomass is one of the key, source. Also, people are trying to use this algae, basically, algae production for the CO2 capture. So again, if you're able to provide, CO2 in a better form, let's say nanobubble form, so one can use this CO2 nanobubbles in terms of photobioreactor. So there are the applications. Of course, membrane fouling and a lot of applications have been, as of now, identified. And many of them, many of the applications I'm working, my research group at IIT Ropar is working in this area. So I will look at some of the application. I will try to explore some of the applications in my presentation. So stay tuned with the properties. So now, if I look at the, of course, I'm talking about properties of nanobubble. So how to generate these bubbles? That is also important topic. So there are various methods by which one can generate nanobubbles. The one method is called hydrodynamic cavitation method. The other is called acoustic cavitation, electrolysis, and membrane method. So there are various techniques by which one can generate nanobubbles. But not all of the methods are can be used for the large scale applications, for commercial applications. For example, acoustic cavitation method can be only can be only used for lab application. It cannot be uh, you know for commercial application cannot be used. Similarly, electrolysis application is something you cannot electrolysis method cannot be really generate a huge amount of bubble, so that this can be also not used for commercial applications. The only the cavitation based method and the membrane method, they are the techniques which can be really can really be applicable for large scale applications. Right now, if I look at the how to detect, how one can measure the nanobubbles, that's also important. Properties. Important methods are available by which one can capture the nanobubbles. So one is called NTA method, nanoparticle tracking method, by which one can detect the nanobubbles. So, for example, I am showing the videos here. So these bubbles can be seen by a microscope fitted in a very small QITS kind of system, where we are passing the laser light, and based on the scattering of light, one can measure, right? One can measure the scattering intensity, and thereafter the Brownian motion of the of the bubble, right? So let me try to show you this video. I'm sure it's not running. So let me look at how this can be. Yeah. So here you can see clearly the Brownian motion or the bubbles under this system, the NT system. So one can clearly observe the Brownian motion. They are under random motion. And that's what I was saying. These bubbles do not rise because of neutrally buoyant bubbles. These bubbles are suspended in the water, and it never rises in the liquid. So that's the important property of the bubble, right? And one can detect the bubble by NTA method. One can also, characterize the surface charge of the bubble by a dynamic light scattering method. And of course, if you pass a laser light, one can clearly see the scattering of light. So there are two images I'm presenting here. The one is without bubble, and with and other is with bubble. So one can clearly see there is a light scattering happening in case of nanobubble, and there is nothing in this case actually, right? So this is what it happens, when you pass a laser light with the in the nanobubble sample, and with there is no bubble sample, right? Now the major question that people normally ask in academics, why bubbles are stable, why nanobubbles are stable. If you look at carefully the micro bubble and macro bubble, it normally bursts actually, right? It's not stable. The question is that why nanobubbles are stable. If I use the classical theory, Epstein-Plaslet theory, old theory, classical theory, so it seems that nanobubbles should not exist for microsecond. If I try to estimate the lifetime of the nanobubble, they are going to be, with this formula, if I try to estimate, it is going to be microsecond. So clearly by theory, if I go by the classical theory, bubbles would not exist. But why bubbles are stable? But if you look at our experimental data, if you look at our experimental techniques by which we have measured, So they are stable. I have already shown the proof, right? I have shown the proof here actually. See, the bubble edges, they are stable. We are able to measure, right? We are able to measure the bubble Brownian motion, which means the bubble exists, right? So the question is that why they are stable. There are several answers. We also have proposed our own theory. So in 2017, 2018, during that time, my research group has proposed a theory by which bubbles stability can be justified, right? So there are several theories. Many of them are not really a correct theory. What we have presented at the time, there are surface charge, and the surface charge basically, negative charge, basically the ions present on the surface of the bubble. These bubbles are really responsible for making bubble to be intact, right? So these bubbles are really stable just because of surface charge, right? So surface charge at the interface basically stops the bubble to further shrink because they act at the opposite of the surface tension. The surface tension and the surface charge both, they act vice versa. They act opposite to on each other, and that's why these bubbles are seen to be stable, right? So what typically studies in my research group we are doing, we are trying to provide the existence of bubble in alcohol as well. So recently we have done a lot of work, a lot of research work where we are providing the proofs why the bubbles are stable in alcohol. I understand, I mean, in case of water, it's fairly fine because there are some certain charge or the OH minus charge is present in the water. But alcohol is non-polar molecule. So do you think the nanobubble can exist in the alcohol as well? So these are the fundamental questions that we are trying to answer in my research group at IIT Ropar. Of course, is it possible that nanobubbles are colloidally stable? So the question is that is possible that nanobubble alone may not be exist, but is it possible that nanobubbles exist just because of there are plenty of other bubbles? So other bubbles present in the system is basically providing a collective stability, or we can say colloidal stability. Is this the case that we've also inquired, that we have also studied? Similarly, we have studied the mass transfer characteristics of the bubbles. Is really mass transfer of the nanobubbles? I mean, if I compare with the conventional techniques by which the mass transfer is estimated, are these nanobubbles are really providing much better mass transport? Apart from that, as I said, is really particle-particle interaction. And interparticle interaction is really, or bubble particle interaction are really going to be improved by nanobubbles. So these things are the fundamental questions that we are trying to answer with the several research papers that were published in the past recent days. Actually, we also have given some sort of a work on electrochemically active nanobubbles, advanced oxidation techniques by using nanobubbles. We're also working on CO2 reduction and hydrogen production by using nanobubbles, sterilization and disinfection by ozone nanobubbles. So bacteria can be killed, virus can be killed, disinfection is possible by using nanobubbles. So these things we are doing, here, at IIT Ropar. So some of the results I will discuss on the existence of the nanobubbles in the in the alcohol and then the water. So what we find actually, yes, in alcohol as well. If you look at carefully the alcohol system as well. So pure alcohol also generate bubble actually, and we are able to see the bubble behavior. Tyndall effect were observed, and we have also measured the bubble dynamics in a pure alcohol, and we find that a bubble also exists in the pure alcohol. So we have measured again. We have proposed again theory. The reason is again the charge actually, despite the fact that the alcohol doesn't carry any charge, but still there is some charge present in the bubble that we have measured. And that's the reason that bubbles are seen to be present in the alcohol as well. We have also, given the evidence, right? People ask, okay, you've given some sort of evidence that is not very scientifically strong, like measuring bubbles, by using NTA machine, by NTA technique, may not give, sound justification that they are bubbles. I mean, that would be particle as well. They are, I mean, they're not necessarily that the thing we are presenting may not be bubble actually. They could be a particle as well. So to verify that those theory that I mean, people are asking question, measuring, recording the videos may not be necessarily that they are bubbles actually. We have developed one other technique is called a refractive index method. So normally refractive index method is basically a very strong proof that if bubble is present, bubble normally should have the refractive index around 1, right? Particle will have the refractive index of more than 1.33, or right. So at least if the refractive index we're getting is around 1, then we can say that they're bubbles. And that's what we have provided proof that we have developed a technique by which by using the Mie scattering theory, we have measured the scattering intensity of the bubbles, and based on the Mie scattering fittings, we have estimated the refractive index, and we find that bubble really has a refractive index of 1, right? And that is the major thing actually. We have developed several theories, as I said, if the bubble are stable. Of course, if the bubbles are stable, this would predict the stable size, and we are able to predict the stable size of the bubble. This is the experimental data. We have monitored the data of the nanobubble density and the mean size. We find that bubbles are stable up to 1 year even, right? So if you see the mean diameter is not changing, the mean diameter is more or less constant. It's not changing much, but the concentration of the bubble is decreasing drastically over the period of time. So which means the bubble is decreasing in number, but still stable, which can be seen by the mean bubble diameter of the nanobubbles. About the colloidal stability, bubbles are colloidally stable, and we have seen, we have measured the colloidal stability. We have measured the barrier. So in case of alcohol, in case of different kind of alcohol, we have measured. So if we go at higher carbon chain alcohol, we find colloidal stability. But if we go lower scale, lower size, if you go higher size, you will not find the colloidal stability of the bubble. Similarly, in case of water, we have also seen in case of water, definitely in the water medium, the bubbles are seen to be colloidally stable. And the barrier that we have seen, right, is very, very high. Energy barrier that we have seen is 625 kBT, which is considered to be much higher compared to the, the colloidal stable. We can say the bubble is colloidally stable because the energy barrier is quite high. Now, the mass transfer characteristics is also very, very important, which we need to understand. So why mass transfer is important? Because if you look at the mass transfer rate, chemical engineers normally know the mass transfer is basically given by KLA, right? So KL is the mass transfer coefficient. A is basically called interfacial area, and the interfacial area is basically given by surface to volume ratio, right? In case of nanobubble, if I look at the interfacial area, right? If you look at the interfacial area for any spherical bubble, it comes around 6 by D. So smaller the diameter, more the surface area, right? So here the diameter of the bubble is much lower. The diameter of the bubble is going towards the nanoscale. So clearly the interfacial area becomes very high. If the interfacial area becomes very high, and that's why the KLA becomes much higher. And that's what we have seen. So in case of mass transfer coefficient, the bubble, in case of nanobubble, the mass transfer rate is seen to be a function of gas flow rate. So if I increase the mass transfer, gas flow rate, we have seen the mass transfer rate also increases. At the same time, the oxygen transfer efficiency reduces because the bigger size bubble you'll create when you are increasing the flow rate of the gas. Several theories we have proposed for the we have measured mass transfer rate, and we have done a lot of other experiment like measuring SOTEs, oxygen transfer efficiencies. These are the typical characteristics of the mass transfer using nanobubbles, right? Of course, the mass transfer rate depends on a lot of other parameters like oxygen flow rate. It also depends on the membrane pore size and various other conditions. So these all have been discussed thoroughly in our recent paper in 2024. You can go through the paper for more detailed information, right? So now I will talk about the alcohol mixture. So let's say we are dealing with the alcohol water mixture where the mass transfer rate to be determined. So if you would like to determine the mass transfer rate in water alcohol system, so it depends on the water alcohol mixture. So it depends on the percentage volume by volume ratio of alcohol and water. If you are increasing the alcohol content, the mass transfer rate has been seen to be increased in case of methanol water mixture. We also seen that the interparticle bridging, or we can say the interparticle contact, has been seen to be increased in case of nanobubble. So what it does actually, nanobubble basically attach on the surface of the particles. For example, if you see in this figure, see these are the particles and these are bubbles. So bubbles basically are here on the particle, and it forms a bridge. So it forms the particle capillary bridging between particle to particle, and this capillary bridging is basically responsible for collection of the particle, right? So that's what it happens actually. And if you look at the real experiment, I will show you here, right? So this is the left-hand side figure. If you clearly see, the left-hand side figure is basically there is no nanobubble present, and in the right-hand side, if you see, there is bubble present. So you can clearly see that there is a particle aggregation, and the particle aggregation is basically happening just because of the presence of nanobubble in the system, right? Again, as I said, mechanism, basically simple mechanism is basically if there are two particles present, the bubble basically forms a bridge, and if the bubble forms bridge, that bridge is basically continuously forms between the various particles together, and in this way it forms a bigger particle from this smaller particle. We have done a lot of experiment on flotation as well, how to recover the fine particles. So we find that the collector doses and collector required during flotation can be reduced, but because the collector also has the same properties, the collector also provides hydrophobic surfaces, and the particle aggregation becomes prominent. So what we have found in this studies, Neelkanth has done a lot of experiment. So what we find that the total recovery of the particles, if I use flotation, so in the microflotation setup, if you perform the experiments, the flotation recovery can be enhanced drastically by using nanobubbles. And of course, the amount of chemical required, recovery amount of the collector doses required has been seen to be reduced drastically by using presence of nanobubble in the system. Next topic, I will elaborate on electrochemically reactive nanobubbles. What we find that if you generate nanobubble by using electrochemical method, these bubbles have been seen to be electrochemically active. And if we are able to create bubbles by using electrochemical method, these bubbles has a property that it automatically reduces the dye. It automatically degrades the dye actually, right? So we have done a lot of experiment on this, and we have seen that the Bubble generation is there, of course, with the time. If you look at carefully, if you increase the time, you'll get more and more bubbles. But these bubbles are seen to be electrochemically chemically active, and it reduces the dye molecule present in the water. Advanced oxidation, we have done a lot of experiment on advanced oxidation, how the tetracycline and other compound can be degraded by using nanobubbles, and dye molecules, of course, can be degraded very easily. And of course, the tetracycline can be also degraded very easily. A nanobubble has advantage than the microbubble because nanobubble is very, very small in size, and it can transfer the ozone and very effectively in the system. As I said, it generates free radical, and we have measured the free radical generation by using ESR measurement, and one can clearly look at the pathway by which the degradation of the dye molecule occurs. You can clearly see the red constant here. The tetracycline concentration. Tetracycline is basically antibiotic, which is often seen in the pharmaceutical industries, and tetracycline can be degraded very easily in a much better way actually. And the rate constant can be seen clearly that, and also the mass transfer can be seen clearly. I mean, it's much better in case of the nanobubbles, right? So there are a lot of studies we have done in case of ER&BS as well. So I'll skip these studies. I mean, you can see the dye and degradation. So how the dye can be degraded. So a lot of study we have performed in the recent days. So what I will do actually, I will show you some more practical studies that we have done in case of a real trial, right? So I will show you the data on pond treatment, right? Let me, so for example, the real application where we have deployed the bigger scale, large scale nanobubble generators to the agriculture applications, where we have deployed bigger machines, and we have measured the plant growth. So you'll see the impact here. So this is the nanobubble effect. So left hand side you see without nanobubbles, and the right hand side you see with nanobubbles. You can clearly see the root growth is much higher, much bigger, right? And similarly, if you look at the length of the root is also much bigger in case of nanobubbles. And similarly, we have seen in case of the number of leaves, fresh weight, dry weight, and shoot length and root length, is all are basically have been seen to be much higher in case of nanobubbles. Similarly, we have done a lot of studies on tomato. Tomato growth is also seen to be much bigger. I mean, the production of tomato has been seen to be increased by more than 20% by using nanobubble treatment, right? And you can clearly see that the root growth is much better using nanobubbles. Of course, the seed germination is much better. One can clearly see that by using nanobubbles, the growth of the seed germination is much faster compared to the a plain water. Several pilot studies we have done in case of treatment of the pond and lake. You can clearly find the results here. We have done 0.5 acre pond treatment before and after. You can clearly see that BOD, COD has been drastically reduced by using this nanobubble technology. Several results we have proposed, several results we have shown here, and one can clearly see even the E. coli can be drastically dropped by using nanobubble. Here we are using a bit of ozone nanobubbles to decrease the E. coli fecal coliform, right? There are similar studies we have done in case of various village ponds. We can clearly see that BOD, COD has been drastically reduced over the period of time. So these are the typical studies we have done in case of pond and lake treatment. There is one study we have done in case of Udaipur. So Udaipur is basically Rangsagar Lake is a big lake. It's 20-acre lake, and here we have done a lot of studies. If you see carefully, let me show the video if I have actually. So let me check at the yeah, here. You can clearly see how this condition of the lake, the entire lake was there actually. So it was completely, filled with the water hyacinth and a lot of other things actually in the system. And after we started using nanobubble generator in the lake and pond, you can clearly see that no just chemical, just nanobubble generators. You can see the DO level was increased from 0. In the beginning, the DO level was seen to be 0. And after installation of nanobubble generator, you can clearly see that it goes around 8 ppm or more than 8 ppm, and that is the biggest thing. If you look at now the condition of the pond or the lake, you can clearly see that it's much better actually. It's there's no algal bloom. There is very fairly clean water. You can see we can clearly see in the entire lake. Right, this is what the condition of the lake is currently now. So yeah, smaller the size of bubble, but the impact is big. You can see the bigger impact of the this technology. So with this, I'd like to conclude my presentation. So the major outcome in case of lake and pond, we have seen there is no algal growth, foul odor was removed. The DO level was reduced, increased drastically. The BOD COD parameter was reduced drastically. No mosquito breeding was observed after the treatment. There is no risk of the waterborne diseases in this case, right? So that's what. So from the lab to the real application that we are doing at IIT Ropar. So it's fully fledged lab and much better lab we have available at IIT Ropar. So we've done a lot of PRO works and we have a lot of installations. So a lot of news we cover in a media actually. So in this way, I would like to thank Dr. Anurag arranging for this talk. I think we have done with the presentation. So we have a lot of collaboration all across the world. People come from Japan, people come from the Germany and here and there. So a lot of collaboration with the people all across the world.